Hello, let's start with chapter 12. Okay, some of you that were in class, this should just be a review. We're talking about some misconceptions about genetics before Mendel's time and why peas was such a good choice. So they thought there were these little, this is a homunculus, right? This little man inside a sperm. That's not, spoiler alert, that's not what happens. So there were a lot of misconceptions, mostly thinking of it as just like mixed paint, just like random particles. And there were really good reasons why the pea was a great choice, and some of the monohybrid crosses that we are going to get to. Okay, so Mendel comes in, lived on a family farm. That's why he was so good at growing peas, and he's our favorite genetics monk right there. Okay, why peas was such a good choice? These are true breeding. Okay, so you either have a round or a wrinkled. You don't have an in-between. You have a yellow and a green. You don't have like a greenish yellow. So this made monohybrid crosses really easy. And these are all the pea plant traits that Mendel looked at. Okay, so let's get started. Each pea, also they had a short generation time, right? So you could grow a pea and fertilize it with itself, and it made a really great test subject. They were cheap, they were readily available, and they were easy to grow in the greenhouse. Okay, some clarification on vocabulary. Pea is the parental generation. Okay, so we're always going to talk about peas, and typically we're going to say they are true breeding, means they are big P, little p in this case. Okay, so the parents, we're going to, unless noted, and say true breeding, they are probably going to be homozygous for something, either homozygous dominant or homozygous recessive. So you have your parent generation, then you have your first generation, your F1s. Okay, this is the offspring of the parents. Then you have your second generation, your F2s. This is the offspring produced by mating the brother and sister. This is in plants. We do this in plants, okay? It's not humans. Settle down. Okay, and then so forth with the generations that go after that. Also, in plants, we're not doing this in humans. Siblings, brothers and sisters. Cross siblings are matings between the brothers and sisters. Okay, back cross is a mating, it's plants. It's plants. Don't judge the plants. This is the offspring mated with the parent. Okay, plants. Say it with me. It's plants. It's fine. And self-cross is crossing a plant with itself. So selfing, here's the pollen, right? And it will go on the stigma and it will cross with itself. This is great because this is how peas work, and this was how Mendel was able to figure out a lot about genetics. A back cross, again, mating of an offspring back to a parent to determine what its lineage is. True breeding, when it says true breeding, the parents have the same traits always. So they're never going to be heterozygous. If it says it's true breeding. Okay? So, again, parental generation, F1 generation, F2 siblings. Okay. Let's talk monohybrid cross. So, true breeding purple, let's say big P, little p, little p. All these are going to be big P, little p. Right? And then all of a sudden, you're going to get 3-to-1 ratio where there's going to be a white one. Let's look at the squares. Okay, Punnett squares. We're going to A's now. Okay, big A by little a. Okay, you're always going to get all of them are heterozygous. Okay, so right, all of them are heterozygous in the F1 generation. Now, we're going to mate the brothers and sisters because they're plants and it's fine. You're going to get a 1-to-2-to-1 ratio. Or in this case, we only care if it's purple. All three of these are purple, and this one is white. Okay, so 3-to-1 phenotypic ratio. 75% will be purple, 25% will be white. We bring in this A down here, A here. Little a here, little a here. Then you bring them across. A here, little a, little a. Get a piece of paper, or better yet, print off the worksheet that I posted on Canvas if you were not in class. Okay, same thing. F1 has all violet. F2 you are going to see that 3-to-1 ratio come back. Here's again how the gametes work for that. Okay. Dominant. So dominant traits in peas. Yellow is dominant to green. Round is dominant to wrinkled. Green is dominant to yellow in terms of pod color. Inflated or full is dominated to constricted. Purple is dominant to white. In flowers, usually the darker color is dominant because that means it's producing a protein versus it's not producing the protein. Does that make sense? Okay. Again, same thing. We have this 3-to-1 ratio when you cross, right? We crossed big P, little p, little p. We got all big P, little p. Now we're going to get 75% purple and 25% white. Okay? And when I say gametes, we mean alleles. Also, point of clarification. Particles turned out to be genes, turned out to be alleles. Again, clarification. So we have phenotype is what color it is. What does it look like? Is it purple? Is it blue? Is it wrinkled? Genotype is big A, little a, big A, big A. So anytime you're looking at a question, what are they asking? Are they asking me for the genes or are they asking me for the phenotype, the color? Alleles, again, are big A, little a. That's what we say when we say alleles or gametes. Dominant, alleles that mask the others, usually designated with a capital. Recessive, usually designated with a lowercase. Flies are different. There's some other things that are different, but by and large, capitals are dominant. Homozygous. Both alleles for the same trait are the same. This does not mean dominant. Okay, you can be homozygous. This is homozygous recessive. This is homozygous dominant. Very confusing. Very often students will see the word homozygous and assume it's homozygous dominant. Not necessarily. Homozygous just means both alleles are the same. Heterozygous means one of each. Okay, gene, again, we talked about that, is the allele. The loci is the place within the chromosome where the gene exists. Wild type alleles happen in flies or in things that we have mutated. So we can say normally flies have red eyes. That's the wild type. That's what's normal. And a mutant allele is rare, something that we know that either we have done or is rare, but we don't do that for most things because it's rude. Unless you're an X-Men and then it's okay, a reciprocal cross. This is a cross where a previous cross is repeated, but the parents exhibit this. So this, again, it's plants. It's fine. We're just looking at whether there's going to be a difference where the female has the characteristic versus the male. So these are reciprocal crosses, right? Does it matter if the female has the trait or the male has the trait? That is a reciprocal cross. These are just vocabulary. Okay, let's do a monohybrid cross, right? So we've got big P, little p, big P, little p. We're pulling this P down. We're pulling this little p down. We're pulling this P here. We're pulling this little p here. Take a piece of paper and do it. Okay. Here we have the cross that we just looked at. This is related to the law of segregation. It says that you're going to pull one of these each time you go through meiosis. That's the whole reason we had to go through the meiosis chapter. This law of segregation is the whole reason. The inheritance hypothesis was not that you just get this sort of green paint. It's not paint. It is at individual alleles and genes. Again, Mendel's first law. Genes come apart during gamete formation. We knew that is the law. Mendel's first law. He formed this test cross, and this is how he proved his law, his first law. Punnett squares. Here's what it looks like. Here's what is the F2 going to look like. We took these two brothers, and we put them brothers and sisters, right? See if you can do it in your head. Here's another one. So we took one of these and bred it back to a parent. Okay, so we've got big P, big P. This is big P, little p, little p, little p, right? So we've got 50, 50. Do that, pause it, see if it makes sense. This is the big P that we brought down. This is the little p that we brought down. This is a little p. This is a little p. 50, 50. Okay, same thing with the Y, right? We're just going to bring these down and you're going to get 50, 50. When you're looking at, we don't know. Actually, here, this is one where we don't know what the parent is. But we got a 50-50 ratio of yellow to green. So what's this parent? It's got to be little y. Otherwise, if it was big Y, we would get this. If this was big Y, we would get this test cross. If it was little y, we would get this test cross. See it? Pause and look. This is if the parent had a big Y here. This is if the parent had a little y here. Trust me. Pause and make sure that you understand that before you move on. Okay. Same thing here. We just did that larger. Okay. Probabilities. This is telling us each time we do one of these crosses, it is independent of the next one. That's how we're going to do dihybrid crosses. Okay, we're not going to make a big giant thing. We're going to remember that each time. So every time you have a child, there's a 50-50 chance of having a girl. If you have 6 babies, the 7th baby, 50-50 chance. It's not cumulative. Every time you roll the dice, you have a 1 in 6 chance of rolling a 6. Okay, and you roll them again, it doesn't matter what you rolled last time. Right? This is why people go into gambling debt, because they don't actually understand how probability works. That's every time you have a baby, it's a 1/2 chance that you're going to have a girl or a boy. So this is how the multiple and simple probability laws work, which we'll get to when we do dihybrid crosses. Okay. Again, both laws can be combined, and you don't even have to you can just think about this. What are the odds of having, let's say you have a cross like this, and what are the odds of you having a white, whatever this is, white pea plant twice? Okay, well, every time is a new time, right? Every time is a completely separate event. So it's a random event every time. Okay, let's think about this one. You are performing a cross involving seed color and garden peas. F1, the first generation, F1s. What F1 offspring would you expect if you cross a true breeding parent with green seeds and yellow seeds? Okay, so that's going to be big Y versus little y, little y. What are you going to get? Pause it before I go to the next one. What are you going to get? You have big Y, and little y, little y. So you're going to get all yellow, right? Because this one only has a big Y to give. This one only has a little y to give. You can do a Punnett square for that if you want. But just know that you're going to take one from here and one from here. You're going to get all of them are going to be big Y, little y. Now, if you bred 2 of those, that's when you're going to get this 3-to-1. Okay, but the answer to this one is 100% yellow. You really have to do a Punnett square, but you can. I would recommend, if you're doing it on a test, to do it that way. Okay. Oh, this one's a little bit backwards. So you have dominant recessive, in which inflated pods were dominant. So inflated pods are dominant over wrinkled pods. You perform a cross and obtain 650 inflated pod plants in your F2 generation. Approximately how many constricted pod plants would you have? So you already know, if you go back here and pretend this is inflated versus wrinkled, you have a 75% chance of getting inflated and a 25% chance, or a 1/4 of a chance, right? 1 out of 4 chance. Can we agree on that? 25% or 1 out of 4. So you have 650. How are you going to do that math? Pause it. Okay, so we already know, here's our Punnett square. We have a 75% chance of getting inflated and a 25% chance of getting constricted, which is a 3-to-1 ratio. Okay, so 1 to 3. You do that here, inflated, you already know that number is 650, so you just divide it by 3. And 217 is going to be the amount of constricted. Think about it, pause it, do the math in your head. Or you don't have to, you can use a calculator, it's fine. Okay, so we did that one. Another one, big R, little r. This is just another one for you to look at, right? We got 50, 50. Incomplete dominance. Okay, snapdragons. Sometimes we have one in between. Instead of being red or white, you have red, big R, pink, big R, white, little r, little r. Okay? So sometimes you're going to have to care about this heterozygous. Okay? That is called incomplete dominance. Still a monohybrid cross. Incomplete dominance is there's just not enough to make red, but there's enough that it's not white. That's incomplete dominance. Codominance is it produces both. It's not a brownish, it's a cow that produces both the red and the white. Sometimes if you look in fur colors of hair colors in horses, you'll see that the horse looks brown, but actually when you get up closer, it has black and white. Sometimes they will look pink. This is incomplete dominance, right? Just because there wasn't enough red to make red, so it's pink. Versus codominance, where it actually has red and white. Okay? That's just a vocabulary difference. Also, we're going to talk about sex-linked real quick. This is when you have to keep track of your X and Y, and it's going to be more likely that the males will have those conditions. Okay, yes. X-linked disorders affect men more because they only need one copy for them to have the disease. So feel free to look at this and write it out. And there are a couple of those problems also. Like this, you're just keeping track of your X's and you're keeping track of your Y's. And you will see half the sons have hemophilia because you only need one copy. Half the sons are unaffected. All the daughters are unaffected, but half of them will be carriers. Okay, that is all on monohybrid crosses.